Okay, so my name is Monique. I am a biology major. I did work under Dr. Snyder this semester and a little bit of last semester. So my presentation is with and without 3-molar fructose and the phycocyanin extractions from cyanobacteria. Limnothrix belongs to the genus Cyanobacteria, and Limnothrix was the culture that we did use for this experiment. They are single-celled organisms that can be found in freshwater and marine habitats as well. They produce their own food. They are able to grow very fast. We was able to witness that in lab as well, which makes it a great culture to use, because over spring break, we did, like he said, add some more nutrients to another artery, and it did, when we came back, it did have some growth in it. It is known for its blue-green color, as you can see in this picture here. Phycocyanin is known for being very toxic, which can cause harm to aquatic animals and also the environment. The phycocyanin is known for its blue color, and it has a wide range of uses, from medicine to food to even cosmetics. Scientists are still trying to find effective ways to extract phycocyanin from cyanobacteria cells. Okay. And also this process is very extensive, and it does cause damage to the phycocyanin levels. The objective of this research was to show that 30% fructose protects phycocyanin from losing its blue pigment during the extraction process, and also compare the extraction efficiency with and without 3-molar fructose. It is hypothesized that due to the increase in velocity of the 3-molar fructose would also increase the amount of extractions that will be needed. And I will get into a little bit of later of why we used fructose. These are the substances that were used. We used fructose, water, and octanol. A little bit about octanol, like it does denature the protein a little bit, but it's at a very slow rate. So that's why when we do have the fructose along with it, it helps as a protective agent. Okay, so we're going to get into some materials and methods here. We did filter the Limnothrix through a Support filter. Once they was filtered, we did put it in a minus 20 degrees Celsius freezer and left it in there for the next day. These were the following solutions that were made. I have my fructose solution, and then I also have the water solution, and these are the measurements that were used. And 2 of those were blank, so one of them was from the fructose solution, and the other one was from the water. Once the solution was made, the 2 halves were, the filters were cut in half, and it was placed into the following solutions. Okay, and then now we have the separation process, and this is the little mini bead beater here. So once they was placed in the solution, it was placed in this little mini bead beater in order to shake and mix up everything, and then it was placed into the centrifuge and spun for 3 minutes at a high speed, and then it was placed into like a little tube, a micro tube holder, to make sure that the 2 layers did not remix. And all of these valves were put through a separation process at least 8 times. And one separation process is just going through a mini bead beater, being shaken up for 30 seconds, and then put in a centrifuge for 3 minutes. All right, and then here for the separation process, so finding a fine tip pipette was used to make sure that we don't get any of that intercellular debris from the filter or any other like dead cells. Only the extractions from the first, the second, and the fourth and the eighth were extracted, and it was placed into a well plate. And then we have the well plate analysis. So we use lost a little train of thought there, sorry. So once they was placed into the well plate, we placed it into the well plate reader to absorb, to scan the absorb spectrum. And from this, the data was recorded, and it was also calculated to make an optimal density graph from those 4 extractions. Okay, and this data here is going to support why we use fructose to begin with. So another student that was also in the undergrad program did this data here. So their experiment showed why why fructose was a good protective agent. As you can see here, she used 4 different substances, and you can see that fructose left you with that same blue pigment which you would like. And then also she had a little graph here that showed the absorbance, and you can see here that the fructose did have a very high spectrum there. Absorbance. Okay, and then here goes into my results that I did here. So as you can see, the fructose versus the water. So doing the first extraction with the fructose, you can see here like not many cells were released. But for the water doing its first extraction, it was like a lot of cells were released, almost to about its max was released. And as time went, it took about 4 extractions for it to meet to some type of like maximum extraction of cells being released. And on here you can see it started to go down after that first extraction. So here it's concluded that the hypothesis was supported by the results, but due to the velocity of the fructose, it did require more extractions in order to release the phycocyanin from the cells. With this newfound phycocyanin, importance for phycocyanin, I think that there needs to be more further research to figure out what is the actual maximum optimal density of that fructose extraction. So online I know it showed where it was like at like 4 was like the highest, but we want to get down to seeing exactly what is the max that it can hold as supportive wise. And that is and that is my references.